Hi, there are different dimensions of biodiversity. To understand inventorying and monitoring of biodiversity, it is essential to understand spatial as well as temporal dimensions of biodiversity. Biodiversity has both the spatial as well as temporal dimensions. I am going to give a brief talk on these dimensions. I am not going to the details of all of this. So, biodiversity spatially it is heterogeneous. There are different types of organism, and between the communities there are changes, and the ecosystem there are changes. So spatially these are heterogeneous, and temporally it is varying over time. Over time the biodiversity will change. It is not a constant value. There is a dynamics there. The spatial dimensions of biodiversity is classified into three: alpha diversity, beta diversity, and gamma diversity. Alpha diversity is the species diversity in a local habitat. It's actually the species richness of a community. How much different types of species are there in a community? That's what the alpha diversity say. So in a community, it's a local area. In a landscape, we may have different ecosystem, and each ecosystem we have different communities. So each community, how many number of species is there? So that's what say alpha diversity. It's actually the species richness of a community, not the whole ecosystem, the community. Secondly, we have the beta diversity. It is a species diversity between two communities. If you are comparing two different communities, what is the species richness or species diversity? If you have two different communities, we will compare the species within these two communities. So the number of unique species which is these communities having is actually the beta diversity. Finally, we have the gamma diversity. Gamma diversity is the overall diversity for the different ecosystem within a region, within a landscape, within a large landscape. So, what is the overall diversity? Overall, all in total, how many different types of species we have? We are not thinking about how much number of unique species, but total number of species, total different kind of organism we are having. I think I will be able to explain alpha, beta, and gamma diversity using this figure. In this figure, each circle, this big circle, represent communities. So here we have three different communities: community 1, community 2, and community 3. And firstly, I am going to tell what is a alpha diversity is. And each of these circles, communities, we have four individuals. But look at this circle. In this big circle, we have green circles and the square one. The green rounds and square, yellow square. So, uh, even though we have the four different individuals in this community, three of them are same and one is different. So, total number of distinct, unique individuals is two. So, it has green round and the yellow square. So, the alpha diversity is two because it has two different types of individuals. Two species, maybe we can say two species. In this circle, if you look, there are three different types. We have the triangle, circle, and square. In this one, four: the star, the circle, the cube, and the triangle. So it is alpha diversity is 4. So alpha diversity is number of different types of species, not the number of individuals. It is not represent the number of individuals. All of this has four individuals, but the types. So that's what the alpha diversity say. Next, we have beta diversity. Beta diversity is always measured between two communities. We can measure the beta diversity between this community and this community. So, how to measure the beta diversity? To measure the beta diversity, what we need to do, compare the populations in or individuals in these two communities. So, I want to find which are the unique species. Unique species means species found only in one community. If the species is found in both these communities, that is not unique. So, in that basis, I am going to find the number of unique species. So, look at this one, the green round. The green round is present in this community but absent here. So, that's a unique species. The yellow cube, which is in this community and this community. So it is not a unique species. So in this community, we have one unique species, that is green round. Come to this one. This one have four different types of species. Out of that, the yellow cube is here and here, but these three, star, triangle and the blue sphere, these three are only present here. So these three are also unique species. So here we have one unique species, here we have three unique species. In total, when comparing these two communities, we have four unique species. So beta diversity between these two communities is 4. If you take the example of this one, just compare it. You just take the green sphere, it is present here but absent here. Then the yellow cube, which is present here, absent here. So when comparing these two communities, it has two unique species over here, and here this one is unique, the triangle is unique, the sphere is unique, but these two are same kind. So here we have three unique species, here we have two unique species. So the beta diversity between these two populations is 5. 3 plus 2 is 5. Finally this one, here just find the number of unique species. Green cube is here but not here, so 1. Blue sphere is here and here, so that's not unique. The triangle is here and here, so that's not unique. So this one has one unique species. Over here we have star, which is absent here, and the yellow cube is also absent here. These two present in both. So here we have two unique species. So here two unique species, here one unique species. So beta diversity between these two communities is 3. Then we have the gamma diversity. Gamma diversity is the number of unique individuals or unique species present in all these communities. So we actually have 6 different types of individuals. The star is here, the sphere is here, the green cube, the red triangle, then we have the green sphere here, and one other unique one is the square, the green square. So totally we have 6 different types of species. So the gamma diversity is the overall diversity. So I hope you understand 3 of these diversities. Alpha diversity is the number of different individuals in a community. Beta diversity, number of unique individuals between two communities, and the gamma diversity is total number of unique species or different species in a ecosystem or a landscape. And when we think about the abundance of the species and number of individuals and the number of species, mostly we will get a log normal distribution. You might have know the normal distribution, the bell-shaped curve, what you are seeing here. So if you just make a plot between the number of individuals and the number of species, so if a species is having less number of individuals, like a species, a rare species, which have only the particular species in birds will be very less, like our hornbill birds, like that, they are very few in number. So if you look at the numbers, you can see somewhere here the number of individuals for some species, the number of individuals is too less. For some other species, the number of individuals are too high, just like the crow. You can find 1,000 crows if you look around for 1 or 2 hours. So some of the very few of the species have large number of individual abundance, and very small number has the less number of individuals in that species. But most of the uh, most of the species have the moderate abundance. So if you look at the any population, any type of ecosystem, the most of the particular species of particular kind will have moderate abundance. Very few are rare, and very few are very abundant. So that's what happens in any population. So that's called a log normal distribution, which is shown here in this graph. What they show is the bird diversity in an island. So if you look at the bird diversity, the island may have 50 types of birds. but most of them are moderately abundant. Very few of them, maybe two or three of very rare birds will be there, and maybe three, four the very abundant birds will be there. So most of them will be at the middle of the curve. So the number of individuals will increase, will be high at the middle. So we will get a bell-shaped curve. So uh, this curve is called a log normal distribution. So this is what we expect in an inventory. If you make a proper inventory, you will get something like that. Another concept is species evenness. Species evenness is relative species abundance. We are in the previous graph, we say the abundance of different birds, therefore how much individuals we are seeing. In relative abundance, we are looking at how common or rare a species is relative to other species in a community. How, if you are taking the birds, basically at the same trophic level, if you are taking birds, one bird species comprise of how much percentage of total birds. So we are getting relative to the whole number, how much is there. So which is the better measure of the species diversity. So it represented the percentage composition of an organism of a particular kind related to the total number of organisms in that area. It basically do in a single trophic level. If you are doing it for the birds, most of them are on the same trophic level. You can do it for the insects of particular kind, or you can do it for the producers. So if you have a type of tree, so out of total number of trees, how much percentage this one comprises of? So that's what say the species evenness. This figure will explain it better. So here we have two different communities. Both of them have 20 individuals. The number of individuals are same. The community A and the community 1 and the community 2 has 20 individuals, and both of them have same species richness. That means the both of these communities have 5 different types of species. The alpha abundance is 5. So here we can see five, actually four, sorry, 4 different types of individuals. You can see A, B, C and D. 4 different types of individuals are here. Here also we can see 4 different types. 1, 2, 3, 4. But when we look at both these figures, even though they have the same alpha diversity and same number of individuals, this one look more diverse than this. It is because of the species evenness. The peculiarity of this community is that all 4 of them is equally abundant. Like A is 25 percentage, B is 25 percentage, C is 25 percentage, and D is 25 percentage. But in this community, A is the most abundant one. Out of 100 percentage, 80 percentage is occupied by A. B is only 5 percentage. C is only 5 percentage. D is only 10 percentage. So this community doesn't have the high level of relative abundance. This relative abundance for A is very high, but relative abundance of B and C and D is very less. So it is less diverse. This one is more diverse. So this community 2, we can say it has more community evenness. So more community evenness is not good for good measure of the biodiversity. When a diverse community should look like this, it is more diverse, more type of individuals are there. Even if both of them have same alpha diversity, the species abundance, species evenness is different. Next, we have the temporal variability. Temporal variability is the variability in population and community over time. The composition of the community always vary depending on the time. Just all the community or the ecosystem has a background variability. In some seasons, the some type of individual number will be increased. In other season, it will be decrease. For instance, insects, maybe it will increase for 3-4 months. Then we couldn't find them. Then after a year, they will again reappear. Or in case of the migratory birds, they will come only in some seasons. So if you measure the biodiversity of a area in that particular migratory season, you will see a big biodiversity. Different types of birds are there. But after 3 or 4 months, it will decrease. So there is always a temporal change. It will change over time. Seasonal variations are there. In the rain season, you can find so many plants, small plants. In summer, most of them might have gone. So the diversity will change over time. There are seasonal changes are there. Otherwise, other than that, there is a background change. Without any reasons, it will fluctuate. The, we cannot, without the interference of humans or any human activities, there is a change in the biodiversity over time. That's called the background variability. So, the background variability is somewhat related to the complexity stability hypothesis. We already learned that if a community is more complex, means if it is more diverse, it is more stable. That's what the complexity diversity theory say. So, if a community with a huge number of organisms or huge different types of organism or very diverse community doesn't have that much background variability. If a community is very diverse, the background variability is less. If the community is not that diverse, the background variability is more. So sometimes the background variability may lead to the extinction of a species. So it is better to have a more diverse community so that the background variability will be less. In the more diverse community, what happened is like if one of the individuals or the type of individuals is decreasing, others may support or complement. So the diversity will be maintained. In small communities or the less diverse communities, that thing doesn't happen. So it is prone to change. So we talked about the species diversity and the dimensions of diversity, spatial and temporal dimensions. Now we are going to jump. We are going to jump into the inventory types. Inventory can be made for species level or the genetic level. Species level inventory says how much different types of species we have. It's basically the number of different types of species. In the genetic level inventory is measuring diversity within a species, within a population of species, how much diversity is. If you really want to measure the performance of an ecosystem, it's better to measure the species diversity or species inventory. We are making the list of all type of organism or type of different species within an area. That's better if you want to know the overall performance of an ecosystem. But sometimes we need to find the whether this particular organism is going to extinct, what is the performance of this organism, how the conservation efforts are, is that doing a difference in the abundance of the organism, things like that. If you are focusing on a single individual or a single type of species, it's better to look into genetic level inventory, how diversity is becoming. So I'll be talking about these two types of inventories in following sessions. Then thank you so much for listening. Thank you. Bye.